Today's notes will cover Chapter 4, the last part of it, where we talk about specialized structures within a cell. So animal cells have some distinguishing features that help you to identify it versus a plant cell. Some of these key features include the presence of lysosomes, centrioles found within the centrosome. So both plant and animal cells have a centrosome, which is where spindle fibers will organize in helping with cell division and mitosis. So both plant and animal cells have that, but animal cells have an additional structure called centrioles that plant cells do not have. The other thing that's on this animal cell diagram that helps to distinguish it from plant cells is the presence of a flagellum. So this flagellum is the whip-like structure that helps cells to move, and some plant cells may have this flagellum, but overall they do not. Notice that there is mitochondria and peroxisomes and the cell membrane and all the other parts like a nucleus, the rough and smooth ER, and the Golgi. Plant cells, on the other hand, have three distinguishing features: the cell wall outside of the cell membrane, a very large central vacuole that stores water and minerals, and the presence of chloroplasts you can see here. So notice that there are mitochondria within plant cells, and students often think that only plant cells have chloroplast and only animal cells have mitochondria, but the truth is that both plant and animal cells have mitochondria. So let's talk about this really special organelle that allows cellular respiration to occur in the cell. So this is the process of breaking down the carbohydrates and releasing energy in the form of ATP. The mitochondria doesn't just have a single membrane outlining its boundaries, but there's actually a double membrane. So there's an outer membrane which is touching the cytosol, and the inner membrane, as you can see here in this diagram, is highly folded. So there's a lot of surface area, and these folds that are found on that inner membrane of the mitochondria are called cristae, and there's a lot of enzymes that are embedded within that inner membrane, and it allows a lot of chemical reactions to occur, and all these folds increase the surface area to increase ATP production. The inner part of the mitochondria is a fluid-filled area called the matrix. There's a lot of enzymes and a variety of chemical reactions happening there as well. And one notable thing that I want you to notice, or actually two, is within the mitochondria, you should notice little dots. Those are ribosomes that are found inside the mitochondria. And so ribosomes are going to be used to produce proteins, but you might wonder, where are the instructions coming from? Well, the mitochondria actually has its own DNA. As you can see inside there, the DNA is not a linear form, but a circular form. So there's a handful of genes that are found in this circular piece of DNA within the mitochondria. Those genes are transcribed into RNA, and then that RNA is read by those ribosomes that are within the mitochondria itself to be able to produce those proteins needed for cellular respiration. Chloroplasts are the site of photosynthesis within plant cells only. The chloroplast also has a double membrane. So you can see the inner membrane and the outer membrane. In this case, the inner membrane does not have all the folding as the mitochondria, but there is another structure within the chloroplast that does have a lot of surface area. So we haven't lost surface area. The main part that you really want to focus on are those green stacks that you see within the chloroplast. They contain a green pigment called chlorophyll and other accessory pigments that give you the reds, oranges, yellows, and purples that are often found in plants or plant cells. But the predominant pigment is chlorophyll, and you see a lot of surface area in all that membrane. So those stacks are, those coin-like things are called thylakoids. Those are the discs, and then a whole stack of them is called a grana or granum is one stack and grana is multiple stacks. And the fluid surrounding those stacks, the grana, is called the stroma. So in mitochondria, the fluid is called the matrix. In the chloroplast, the fluid is called the stroma. Now, two other things I want you to notice that are inside the stroma of the chloroplasts, and that is the presence of a small circular piece of DNA. So chloroplasts have their own DNA that's separate from the nuclear DNA, and it has its own ribosomes in there. So you can imagine there's going to be the reading of the DNA to transcribe into RNA, and then ribosomes will read the RNA to make proteins that are specific for processes that happen within the chloroplast. Now you might be wondering, I didn't know that there was DNA and ribosomes inside the mitochondria and chloroplasts. Well, that is a unique feature of those particular organelles, which has led scientists to come up with the endosymbiont theory that mitochondria and chloroplasts share a very similar origin in the evolutionary history of life. So the first organisms that existed on Earth were prokaryotes, and scientists hypothesized that there were photosynthetic prokaryotes that got engulfed by other prokaryotes and then lived in a symbiotic relationship within those engulfing cells. So that's how you get the term endosymbiont, because it's a symbiosis relationship, but endosymbiont is that it lives within the cells itself. And so what is some of the evidence that supports this particular theory? You can see that the diagram shows that there is an ancestor to a eukaryotic cell, is like the host cell, that has some kind of nucleus structure, and then engulfed cells that were non-photosynthetic prokaryotes that became a mitochondrion. And so if this like pre-mitochondrian structure or cell existed on its own before being engulfed, it makes sense that it would have its own DNA and ribosomes. And then when it got engulfed, the inner membrane was the original membrane of this prokaryote, and the outer membrane is the host cell's membrane as it got engulfed. So you see the presence of two membranes. And then at some point in time later on, this non-photosynthetic eukaryotic cell then engulfed a photosynthetic prokaryote. So that photosynthetic prokaryote, as you see in this diagram, it has its own cell membrane and inside its own circular DNA and ribosomes. And then when it got engulfed, its original membrane becomes the inner membrane and the host cell's membrane becomes the outer membrane. The double membrane structure of just these organelles alone suggests that there is ample support for this endosymbiont theory. The other thing for you to know is, besides the double membrane structure, each of these organelles have their own ribosomes and DNA, is that within the cell, both the mitochondria and chloroplasts can reproduce independently within the cell. And they reproduce in a way that looks like how bacteria or other prokaryotes would reproduce through binary fission. So they don't reproduce like the other organelles, they reproduce independently. And then when this cell divides, it randomly sort of divides out the mitochondria and chloroplasts into the daughter cells. Another interesting organelle are peroxisomes. These are organelles found in both animal and plant cells, and the function of them is to break down fatty acids, detoxify alcohol, and in the process of doing so, this involves the production of hydrogen peroxide. Now, hydrogen peroxide is actually toxic to cells and can, eat away at cells, which is why you put hydrogen peroxide on cuts or the surface of your skin to kind of clean and disinfect it. That's because it would be killing bacteria and actually is kind of killing some cells too. So there is a enzyme called catalase that will further break down peroxide into safer components like hydrogen, water and oxygen. Now, cells are held together with its own structure called the cytoskeleton, and it's a network of protein fibers that are found within the cell. And its function is to support the cell, give it some rigid structure, motility. Some of these fibers will be able to contract to help cells move, and also move organelles within the cells. And these fibers also help to regulate biochemical activities. So these fibers have three major sizes or categories. The largest type of fibers are called microtubules. The smallest ones or thinner ones are called microfilaments. And in between those two sizes is intermediate filaments. So if you're wondering how organelles get moved, they actually move by proteins that will actually help to move, like for example, vesicles along a network of microtubules, and they'll actually be walking along these microtubules to move some of these organelles. Underneath the inner part of the cell membrane is microfilaments, and they help to maintain cell shape. So plant cells have some additional structures that are different than animal cells. Besides the cell wall, which helps to protect the plant and maintain its shape, there's also little channels that link together the contents, the cytoplasmic contents between two plant cells. These little channels are called plasmodesmata, and they allow the passage of small molecules, things like glucose, amino acids, water can go between two cells, and that allows materials and even hormones to be transferred from cell to cell without having to be exposed to the exterior environment. Now, the cell wall is composed of type of carbohydrate called cellulose. As we've talked about before, we are unable to digest cellulose, but it's important to eat plant material in your diet to get that fiber that comes from plants. Now, animal cells have something called an extracellular matrix, or the ECM, and this is structures that are found on the outside of cells. So therefore, you get the name extracellular, outside of the cells. And as you can see, it contains a lot of fibers and that are composed of glycoproteins. In the diagrams, anything that's a protein is going to be shaded purple. That's the color coding that Campbell uses. And you can see these fibers that are really long that's out there. And collagen is an example of a glycoprotein fiber that helps to maintain the structure on the exterior of the cell. So on the interior of the cell, you have microfilaments here that are in the orange, and on the outside of the cell, to hold together multiple cells on the outer layer, is these glycoproteins like collagen. And the function of the extracellular matrix is to strengthen tissues and transmit external signals. So some of these proteins that you can see have extensions coming out of them, and some of the proteins may have extensions that contain polysaccharides, and they help for cell signaling and in terms of for immune systems to recognize cells. So some of those polysaccharides on the surface of the cells help to identify what are your own cells versus what are foreign cells. Now, two animal cells are joined together usually in a tight fashion, and there are three different ways that animal cells are joined together along the cell membrane. These are called intercellular junctions. Tight junctions are when two cells are fused together to form a watertight seal. You can imagine that's probably happening within the surface of like your skin cells, for example. And so there are proteins that have what looks like a quilted effect. So quilts are held together. There's multiple layers of fabric, right? And the quilting allows to sort of strengthen and bond together the multiple layers. Same effect with tight junctions in animal cells. Desmosomes are the rivets. So some of you may have like a pair of jeans, and the jeans often have these little metal button-like things because the material that jeans are made of can be kind of tough, and so you might need some sort of these like metal buttons to kind of hold them together. At least in the olden days they did. So you can see that desmosomes are in this picture, and again they're also proteins, and they help to fasten adjacent cells together so it forms a really strong sheet. The last thing to note is that there are some channels that go in between two animal cells, and these are called gap junctions. And and small molecules like ions and sugars and can pass through. So plant cells have the channels called plasmodesmata, but animal cells have the gap junctions. So this slide summarizes the features of plant versus animal cells. Plant cells have central vacuoles, chloroplasts, cell walls, and plasmodesmata. Animal cells have lysosomes, which plant cells don't have, centrioles within that centrosome region, flagella and cilia for motility and motion, desmosomes, tight junctions, and gap junctions, and then the extracellular matrix. That is on the outside surface of animal cells only. The last thing I want to encourage you to do, now that we've covered all the major organelles and parts of the cell, I want you to go ahead and look up the video, The Inner Life of the Cell. It was produced by a Harvard video team and has beautiful music and graphics, and see if you can identify the various organelles as you watch this video.